Good morning. We're going to get started in just a minute. Could I actually ask each of the speakers to come up to the stage? We're gonna seat you in order, and we're going to make this as sort of collaborative as possible. So Doctors Pisano, Shelby, Kohler, So, and Navoli, please. So why don't we get started? It's 8:30. I'm David Hackam from Johns Hopkins Children's Center and Johns Hopkins University. And actually, I'm going to sit there, but we've got one right at the very end. Don't worry about that. There's one right at the very end. Thanks. And let's just make a little more room if we need to. That's great. And on behalf of my co-moderator, Dr. Agostino Pierro from the Hospital for Sick Children and University of Toronto, we'd like to welcome you to the research session, which is the product of the research committee on the topic of necrotizing enterocolitis. Just a little bit about the session, and then I'll launch into the tribute, and then we'll start the session. So the research committee was tasked with developing a program as part of this year's meeting, and we decided to focus on NEC, and we really wanted to make this as collaborative and interactive as possible. But rather than have Dr. Pierro and or myself discuss our research in the field, we elected as a committee to showcase the work of the trainees that are working in this really exciting area. And rather than select from submitted abstracts, since all the abstracts were so outstanding, we selected them all. And in order to allow for discussion around the various themes that will be raised by each of the talks, we thought we would invite each of the speakers to stay on the stage throughout and even to discuss your colleagues' work. And I also see some luminaries in the field in the audience, in Dr. Besner, in Dr. Hunter, and others. I see Dr. Helmrath just walked in and others. And so our goal is to create an environment today for discussion and shared knowledge. So that's what we hope to achieve. Does that sound good? Now the tribute. So I don't have a fancy schmancy Hollywood-style video with a soundtrack and plot twists and endings and dragons and fire, because this is a tribute about Julius Lister, who actually I had trouble finding anything about until I really dug deep and realized that Dr. Lister and his history is really the history of APSA. Can I just, by show of hands, does anyone in the audience know Dr. Julius Lister? Hello, sir. I think that's sir. Thank you. And so if you ask people if they recognize the names of some of the other luminaries that have been tributed, even today, they are sort of household names in pediatric surgery. And Dr. Lister's name is not one of those, but I really think it should be. My source material is the source material of 2019, which is Facebook, Twitter, and LinkedIn. And I got nothing. And so then I went to the source material of 2002, and I spoke to people that may have actually worked with Dr. Lister. And there are two tremendous surgeons, and I thank them for their comments. Dr. Lister trained with C. Everett Koop in Philadelphia. Dr. Koop, of course, would go on to be the Surgeon General of the United States under Ronald Reagan, and became a pediatric surgeon that was highly regarded, who was highly regarded as a tremendously professional, highly regarded, and actually adored general surgeon who operated exclusively on kids and developed a robust practice in the Boston area and was widely acclaimed and beloved by his patients and their families. That was the story from one surgeon, and really I could have stopped there, because what better tribute to a colleague than the adoration of your colleagues and your patients. Mark Rowe gave me a little more insight. Mark Rowe, of course, was the Surgeon-in-Chief at the Children's Hospital of Pittsburgh just before I trained there, and he told the story of APSA through the eyes of Dr. Lister. Dr. Lister had trained under Chick Coop, and at a time when pediatric surgery was developing as a specialty. At the time, there were maybe 250 pediatric surgeons, and they were all associated with the AAP, and a group of young pediatric surgeons, according to Mark Rowe, decided to break off and form their own organization and hold an annual meeting at the Chicago Hilton Airport. And these were the so-called Young Turks, the young surgeons, of which Dr. Lister was a founding member. This organization had difficulty gaining traction. The senior members of the discipline had no interest in these young surgeons, and in fact they were being largely talking to themselves until they had the brilliant idea of inviting Dr. Gross to be the president. And once Dr. Gross became the president, the organization took off and literally became APSA as we knew it today. And as Dr. Rowe said, the original young guys sort of then got left out. Dr. Swenson was one of those founding members who worked closely with Dr. Lister, and we heard a tribute to him this morning. And he and each of those founding members really formed the foundation which brings us together as a group today. And so, in summary, Dr. Lister was a truly accomplished, talented, and beloved surgeon who had the distinction, I would say, of being a pioneer in our field by breaking away from those that came before him and starting an organization to which we are all part of now. So that's my tribute to Dr. Lister. Now NEC. NEC remains the leading cause of death from GI disease in premature infants. And certainly this is a disease that as an organization, I would say, we cannot give enough focus. And I would also say that it is the people on this podium that are going to take this disease and treat it. And so with that, I would like to introduce our first speaker. And this is the first, if we could have the first slide of S69, S like Sam 69. And the talk is entitled Human Breast Milk-Derived Exosomes in the Protection Against Necrotizing Enterocolitis by Drs. Pisano, Albarari, Farrell, and Galley, Wang, and Brigstock from the laboratory of Dr. Gail Besner. Dr. Pisano, please.
Good morning. I would like to thank the APSA Research Committee for the opportunity to present my work. Here are our disclosures. Necrotizing enterocolitis is a devastating gastrointestinal disease that is the leading cause of death in premature infants. Despite years of research, there is still no known cure. We do know, however, of some protective agents, one of those being breast milk. Breast milk protects against NEC to some degree. It contains multiple components that are responsible for these protective effects. However, despite years of encouragement of breastfeeding in the NICU, the incidence and mortality of NEC remains unacceptably high. Many body fluids contain extracellular vesicles, which are bilipid membrane nanoparticles about 30 to 200 nanometers in size, which contain DNA, RNA, and proteins. They are produced by most cell types and may account for some of the therapeutic properties of breast milk. Since breast milk is protective against NEC, and since breast milk contains extracellular vesicles, we hypothesized that human breast milk-derived extracellular vesicles could reduce the incidence of experimental NEC. In the following experiments, rat pups were delivered prematurely by C-section and randomized into the following groups. Group 1, breastfed uninjured pups. Group 2 pups were untreated. Group 3 pups received a single intraperitoneal injection of breast milk-derived extracellular vesicles at a dose of 4 times 10 to the 8th. Group 4 pups were fed a formula containing breast milk-derived extracellular vesicles at a dose of 1 times 10 to the 8th per feed for the duration of the experiment. Groups 2, 3, and 4 then underwent a series of stressors over the next 96 hours, including hypoxia, hypothermia, hyperosmotic fees, and a dose of lipopolysaccharide to induce experimental NEC. Pups were then sacrificed upon clinical signs of NEC or after 96 hours, and their intestinal tracts were harvested. Intestinal injury was graded using a histological injury grading system. Any grade of 2 or greater is consistent with NEC. Here are our histologic injury results. The percentage above each bar represents the overall incidence of NEC, with the red shaded areas signifying grades 2, 3, 4 of NEC. As expected, breastfed uninjured pups did not get NEC. Pups that were exposed to stress but were untreated had a 62% incidence of NEC. Pups exposed to stress but received an intraperitoneal injection of extracellular vesicles had a significant decrease in the incidence of NEC to 29%. Finally, pups exposed to stress that received a formula containing extracellular vesicles had an even further decrease in the incidence of NEC to only 11.9%. Here, we looked at the ability of extracellular vesicles to protect rat intestinal epithelial cells from hypoxia reoxygenation injury in vitro. On the left, we looked at the effects of extracellular vesicles on cell proliferation. Compared to the normoxic control, hypoxia reoxygenation decreased cell proliferation by about 50%. However, adding increasing numbers of extracellular vesicles to injury-exposed cells significantly increased cell proliferation in a dose-dependent manner. On the right, we looked at effects of extracellular vesicles on apoptosis. Compared to the normoxic control, hypoxia reoxygenation injury significantly increased apoptosis. However, treating these cells with 10 to the 8th extracellular vesicles significantly decreased apoptosis. In summary, breast milk-derived extracellular vesicles protect intestinal epithelial cells from hypoxia reoxygenation injury in vitro. They protect intestines from experimental NEC in vivo. They can be given enterally for ease of administration, and they may prove to be an effective therapy for clinical NEC in the future. Thank you for your attention, and I'll take any questions.
So that was beautifully presented, and really exciting work. Thank you. Share a little bit for the group, just a general question about your model. Mm-hmm. One of the questions that always comes up is how representative is the model to the disease? I would say I think your model is extremely representative, but there may be those that take issue with studying in a mouse a disease that is so complex. And secondly, tell us a little bit about what you think the extracellular vesicles are containing.
Where they're contained or where they're coming from?
Well, what's inside them?
Oh, okay. So first I'll go back to the model. So basically the reason that we designed the model how it is because NEC can be caused by many things such as hypercaloric feeding, so sometimes the baby's breastfed and then switched over to formula, and that can cause a sudden incidence of NEC for the baby to have that. Secondly, we do see NEC in a lot of babies that, let's say, have cardiac issues because they have ischemia. Since their heart can't pump blood, the first thing that their body's going to do is not provide the intestines with blood, but rather other organs. So in ischemic models, we can see that those babies get NEC as well. So those are probably the two biggest things. Also, there's talk of NEC having an infectious component to it, so that's why we give the lipopolysaccharide dose as well to represent that component. And then to address the extracellular vesicle question, so these contain DNA, RNA, proteins, just for an example, but they also contain parts of the cells from which they were derived, and most cells in the body do produce extracellular vesicles.
That's great. Thank you. Please.
Augusto Zani, Sick Kids, Toronto. Really elegant study. Congratulations. Two questions. One is along the line of Dr. Hackam's question about if you looked at what your vesicles have inside, have you done proteomics or RNA sequencing analysis?
So we have in our lab, we've started to do RNA sequencing, but not with the breast milk particularly. So one of our scientists is looking at blood and urine derived from infants. So our next plan is to do the same with the breast milk. There have been multiple studies done in the literature on proteomics, and it's really interesting because in these studies, they have found over 600 proteins from the breast milk extracellular vesicles that have different active proteins than the breast milk itself. So this suggests that extracellular vesicles are different from the breast milk overall, and that's why they may play an important role in preventing NEC.
Thank you. And the other question is the question that people that work with extracellular vesicles always ask, which is, does size matter? Mm-hmm. So you showed us, and this is actually what we also recently published, that the more exosomes you give, or extracellular vesicles you give, and the more effect you get. But do you think that the size of the vesicles in your model matters? So if you're giving small vesicles, it's different than if you give larger vesicles.
So yes, I agree that there are a lot of studies showing the more you give, the more effective it is, and I'm currently doing that study to see if there's a significant difference between the amount that you give. As far as the size, I would have to study it more in depth, but from the preliminary studies I have done, I have in the different samples that I give, there are a variety of sizes. So I would say the average particle size in the different samples that I have given are different, and they vary anywhere from about 50 nanometers up to 200 or even more in some cases, and they've all showed the same effectiveness on the decreased incidence of NEC.
Thank you.
Please.
Hi, good morning. Todd Jensen from Connecticut Children's. Really good study. We're doing work with amniotic fluid-derived exosomes, and I'm just curious if you've characterized the kind of cell types you have in the breast milk stem cell population or the breast milk in general, and where the exosomes are most beneficial from in that population.
So my preliminary studies also looked at extracellular vesicles derived from stem cells. So that's kind of how I started this study. And from the breast milk-derived extracellular vesicles, that's still up in the air about where they specifically are from. So recent literature have shown that they could be from the breast adipose tissue, the breast epithelial tissue, and there's even evidence to suggest they come from immune cells. So that would be a really interesting study in the lab to actually grow those cell types and then carry out a series of experiments that way.
Okay, well, Dr. Pisano, thank you very much, and congratulations for your excellent work.
Thank you.
The next paper is from the same group of Dr. Besner lab, and this time the etiology of necrotizing enterocolitis, infectious or inflammatory. And this is presented by Dr. Rita Shelley.
Thank you for the opportunity to present our work. Please note our disclosures. This is necrotizing enterocolitis, or NEC. Despite decades of research, there's still no known cure for the disease, and the mortality remains as high as 30%. The pathogenesis of NEC is multifactorial, with prematurity, low birth weight, enteral feedings, and altered gut microflora all implicated. The focus of our work is on the altered microbiome. We hypothesized that the antimicrobial and anti-inflammatory effects of the probiotic Lactobacillus reuteri contribute to its ability to protect the intestines from experimental NEC. To address some of the limitations of previous probiotic use, we developed a novel delivery system that enhances the beneficial effects of Lactobacillus reuteri by delivering it in its biofilm state, which dramatically reduces the incidence and severity of NEC. To develop this formulation, we start with free-living LR and incubate it with biocompatible microspheres, inducing biofilm formation as shown here. Furthermore, the microspheres can be loaded with beneficial cargoes such as sugar, promoting increased biofilm formation and increasing the efficacy of the probiotic. As stated previously, Lactobacillus reuteri possesses both antimicrobial and anti-inflammatory properties. To determine which of these effects contribute to its efficacy, we previously altered the bacteria to eliminate antimicrobial Reuterin production. We found that this decreased the efficacy of the probiotic. In these current experiments, we alter the bacteria to eliminate histamine production. Here we show how we altered the bacteria to eliminate histamine production. We then utilized an ELISA assay to demonstrate that wild-type LR produces histamine, but the mutant does not. We then tested these knockouts in our experimental neck model. In our experimental neck model, premature rat pups are delivered via C-section and randomized to treatment groups. A control group where pups are exposed to neck but are untreated, or a single-dose treatment of either free-living LR, LR in its biofilm state, mutant free-living LR, or mutant LR in its biofilm state. Pups are then exposed to asphyxic hypothermic stress for 96 hours. Finally, intestinal sections are graded histologically, and a grade 2 or greater is considered consistent with neck. Here are our results, with the red shaded areas of the bars representing grades 2, 3, and 4 neck. Breastfed animals had no neck as expected. Untreated control animals had a 72% incidence of neck. 56% of animals treated with free-living LR had neck. Additionally, 56% of animals treated with mutant free-living LR also had neck. These results were not statistically significant from untreated control animals, demonstrating that these formulations were ineffective. Importantly, of animals treated with wild-type Lactobacillus reuteri in its biofilm state, they had a significantly decreased incidence of neck to 18%. In contrast, animals treated with mutant LR in its biofilm state had an increased incidence of neck to 47%. In conclusion, we have shown that the anti-inflammatory effects of Lactobacillus reuteri contribute to its ability to reduce the incidence and severity of neck. We previously demonstrated that the antimicrobial properties of Lactobacillus reuteri also play a role. These results suggest that both an infectious and anti-inflammatory component are associated with neck. This novel probiotic delivery system may be useful for the prevention of neck in the future. I'd like to acknowledge the NIH for funding this project. Thank you for your attention, and I'll take any questions.
Thank you very much for this beautifully presented and fantastic study that you've done. I could see Dr. Ford already there, ready for question.
Yeah, I'm Ford from Miami. That sounds weird, doesn't it? This is a beautiful study and elegantly presented. Congratulations. Now, to fulfill Koch's postulates, we have to be able to culture the stool to see if indeed the mutant form that you delivered indeed was responsible for the the protective effects that you saw. So what did you see when you cultured the stool? Were you able to actually effectively colonize the intestine with this particular form of bacteria and then isolate it and give it back and see that indeed it is responsible for all of the protection that you saw?
So we have done those studies, and so the key part of our formulation is not only the bacteria, but also the microspheres. And what we saw is that the microspheres are gone by the time of the 96 hours of the experimentation, and we are getting LR that's still present in the stool. One of the important features of the formulation is, of course, we want it to colonize and stick around long enough to make a difference, but we don't want it to cause sepsis and the microspheres clear quickly enough that that's not a concern.
Okay, thank you.
Thank you.
Thank you. Dr. Hemrath.
Congratulations, Helmrath, Cincinnati. Thank you very much. I think it's real important what you all are doing. In establishing a healthy microbiome is the key, I think, in this disease. In a healthy state, I think the black box I'm still looking for is the metabolome. So you speak to the histamine, but the truth is, what is the metabolome of the microbiome doing that's protective? It's going to be far greater than just histamine inflammation. Have you started looking at metabolic profiles prior to your NEC model as an insight of what you're doing metabolome-wise to put the intestine in a state where it can withstand the stress of NEC? Thank you.
Thank you for that question. So we have previously looked at the metabolome of the pups before they were exposed to NEC and isolated some pathogenic bacteria that seemed to appear to lead to NEC. And what we found is that when, as some of you may know, rat pups can be quite resistant to getting NEC. And so when they don't have that bacteria present, they don't develop NEC. And so while we haven't done the exact studies to determine how our formulation is changing that microbiome, we notice that there's a difference in those pups after they've received the formulation.
So when I say metabolome, it's not the bacteria, it's the products they're producing. And so regardless of it's lactobacillus or whatever, there's a symbiosis that's occurring. And I think you have the perfect setup to take these to the next step. And I really think that's a black box that's going to really be the future, I believe, in how we're going to get therapies to treat NEC.
I think you're right, Mike. So that was beautifully presented, as always, and really elegant. The concept of using bacteria as a delivery system is really exciting, and your ability to manipulate those bacteria for health benefit. You chose histamine. Tell us a little bit of the background, how you selected histamine as your target, and how easy is it to manipulate these bacteria to serve us in ways that clearly in this model they are?
Yes, so we looked at several things. A couple of other areas that we looked at was this particular strain of lactobacillus has the ability to form a biofilm. Not all of them do, and that is based in the gene GTFW. And so we've run those experimentations, and what we recognized when we knocked out GTFW, again, we had a decrease in the effect. And so then the next step was to see what else may be contributing to the effect of the probiotic, because as you see in my results, the knockouts, they lose some of their efficacy, but not completely. They don't go back to untreated animals. And so we sort of went in a step-by-step process. The next step is to do a double knockout, because again, there is some suggestion that it's not just the histamine and it's not just the reuteri that are playing an active role in this.
Perfect. Thank you so much.
Thank you.
That's great.
Can I ask you about the timing of your maneuver? This may have an influence on the results in sense of changing the microbiota or changing the inflammatory state. Now, do you think that delivering this bacteria at later stage may have a different response than giving it early on, like you did?
Correct. So currently our experiments are designed so that it's prophylactic. However, the other side of that is that I also work with a Clostridium difficile model, and I use the same probiotic, and I'm currently testing it as a treatment. So the animals already have C. diff, and then I utilize it as a treatment, and excitingly, we're seeing very similar results.
Very interesting.
Great. Well, thank you so much
for
sharing that data. All right. It's great. All right. Really great work from Dr. Besner's lab, and thanks for sharing those results with us. I'd like to bring to the podium Dr. Mark Kohler, I think, for his project, for his abstract entitled, "Is Ileus a Cause as Opposed to a Consequence of NEC? Reversal of Intestinal Dysmotility Reduces the Severity of Experimental NEC Through Increased Neuron Signaling." And in addition to Dr. Kohler, co-authors included Ladd, Fulton, Yamaguchi, Prindle, Wang, Sodi, and your humble co-moderator. Dr. Kohler.
Thank you. Thank you to the program committee for the opportunity to present our work.
Welcome.
It's an absolute privilege to present in front of this audience. Our lab is supported by funding from Abbott Nutrition, Novium Biotherapeutics, and the Maryland Stem Cell Research Fund. So abdominal distension and feeding intolerance are frequently the earliest clinical signs of NEC, and damage to the enteric nervous system has been demonstrated in both humans and animal models of the disease. This is thought to contribute to the long-term intestinal dysmotility that affects many NEC survivors. Further, the ENS of the premature intestine is underdeveloped, and this predisposes to dysmotility. The enteric nervous system is frequently referred to as the second brain because of its dense and complex network of enteric neurons and glia. It's responsible for coordinated peristalsis and also plays a role in maintenance of the epithelium. ENS damage and subsequent dysmotility are generally thought to be a consequence rather than a cause of NEC, and our understanding of the role of ileus in the development of the disease is limited. We hypothesize that because it is frequently the earliest clinical sign of NEC, that ileus and ENS damage are really early contributors in the disease development, and therefore reversal of dysmotility would attenuate NEC severity. To study the disease, we use a mouse model in which 7-day-old pups are isolated from their mothers and housed in a NICU isolette to eliminate the possibility of breastfeeding. Instead, their gavage-fed formula, which is mixed with bacteria that was cultured from a patient with NEC totalis, and they're subjected to brief periods of hypoxia twice daily. At the conclusion of the model, we find a similar pattern to human disease with patchy intestinal necrosis, luminal dilation, and pneumatosis. Disease severity is assessed by survival, weight loss, and gross exam, histologically using a validated NEC severity score which assesses for microscopic damage, and by measurement of gene expression of inflammatory cytokines from intestinal segments. To study motility, we calculated the percent GI transit of a non-absorbable fluorescent dye. We performed transabdominal ultrasound to assess for peristalsis, and we looked at the degree of luminal dilation and abdominal distension. Here we confirm that the enteric nervous system of the premature intestine in both humans and mice is underdeveloped, as evidenced by low levels of staining for the glial marker GFAP. However, rapid maturation of the enteric nervous system occurs in the final stages of development and just after birth, and this expanded enteric nervous system is reflected functionally by increased intestinal motility in the first weeks of life, seen in the graph on the right. The immature intestine, which we know is at risk for NEC, is thus predisposed to dysmotility. Furthermore, mice who are subjected to experimental NEC show damage to the enteric nervous system, seen here by loss of staining for the neuronal marker TUJ1 in green and loss of staining for the glial marker S100 beta in red. The damaged enteric nervous system in NEC is dysfunctional, with decreased intestinal motility. And these findings led us to consider whether early induction of ileus would worsen the severity of NEC. To produce an ileus in the experimental model, we administered the medication loperamide, which is used clinically to treat diarrhea, but actually acts as an opioid receptor agonist in the small intestine to relax smooth muscle tone and decrease peristalsis. These clips obtained by transabdominal ultrasonography show that in control mice treated only with saline, you see normal wave-like peristalsis, whereas the intestine of mice treated with loperamide show no coordinated movement and their abdomens are distended. Functionally, loperamide treatment leads to lower percent small bowel transit. After induction of ileus in mice who were subjected to experimental NEC, the bowel lumen dilates and it fills with gas, and the final result is a more severe phenotype with increased pneumatosis and worse histologic severity score. Additionally, the induction of ileus led to increased gene expression of inflammatory cytokines IL-6, IL-1 beta, and TNF-alpha. So at this point, we had established that the enteric nervous system of the premature intestine is underdeveloped and predisposes to dysmotility, that ENS damage and ileus occur in NEC, and that augmenting that ileus worsens the severity of NEC. In an attempt to reverse this component of NEC pathogenesis, we administered the prokinetic medication metoclopramide, which acts on serotonin and dopamine receptors in the small intestine to increase peristalsis. And metoclopramide administration did, in fact, attenuate the severity of the ileus associated with NEC in our model. As we hypothesized, reversal of this dysmotility led to a reduction in NEC severity as shown by decrease in the expression of pro-inflammatory cytokines and a reduction in histologic severity score. To confirm that this improvement was secondary to the motility effect of metoclopramide, we tested a second prokinetic agent, cisapride, and had similar findings with improvements in motility and reduction in neck severity. Interestingly, in both of the prokinetic models, although motility was restored and severity was decreased, damage to the ENS persisted, demonstrated here by a similar reduction in neuronal staining for TUJ1 compared to breastfed controls. And what this indicated was that although the density of neurons was decreased, the prokinetic agents were able to exert their effect by increasing signaling in the remaining neurons to improve motility. To conclude, these findings demonstrate that damage to the enteric nervous system and associated ileus is an early component of neck pathogenesis, and that restoration of motility reduces the severity of neck. However, restoration of motility alone does not protect the enteric nervous system from damage. The next steps in this line of research will be to better understand the mechanism by which ileus instigates neck, to develop preventative strategies to encourage ENS maturation and improve motility, and to design safe and effective treatments for the reversal of dysmotility in patients who are diagnosed with neck. Thank you to the members of the Hackam Lab and to the rest of our collaborators at Hopkins, and I'm happy to take any questions.
Thank you very much. This is a beautiful presented and also fantastic study that you and Dr. Hackam have done in the lab, and you need to be congratulated for your work. But I could see already important people line up, so let's open a discussion on this very important topic. Please.
Hi, I'm Kathy Hunter from Lurie Children's Hospital, Chicago. As always, just beautiful work from the Hackam Lab. Fascinating results. What I was curious about, and I know that this will be one of your future directions if you haven't started it already, is have you been able to look at the microbial profile that you're seeing in the animals that have the ileus? Because one of the hypothesis that may exist is this a situation where the neck is worsened because there's almost a stagnation, allowing for a longer kind of pooling and interaction with non-beneficial bacteria with the intestine. So that would be my first question.
Thank you, Dr. Hunter, for that insightful question. We are looking at the bacterial profile in the patients who have an induced ileus. I think that the mechanism by which ileus instigates neck is probably similar to the way that many intra-abdominal inflammatory and infectious processes happen with stasis, as you mentioned, bacterial overgrowth, and then distension, venous congestion, and ischemia. So that's going to be a key component to our next experiments. Thank you.
And then my second question was, I really enjoyed your ultrasound. That was neat imaging and fun, methodology for the animals. Have you looked at potentially quantifying the activity of the gut? So you kind of showed it very much like a binary, this is normal and there's very little. I would be interested to see if you could actually quantify the movement of the intestines so that you could almost grade that as if you have this amount, then this is more beneficial versus this is the critical dropping off point of lack of motility.
Thank you. I haven't come up with a method for doing that, but it's certainly something that I'm thinking about and trying to develop. We're lucky at Hopkins to have collaborators who can help us perform these sort of things I had never imagined that we could perform transabdominal ultrasound on a little mouse pup. But we use the quantitative GI transit of the non-absorbable fluorescent dye in order to make sure that we had sort of quantitative data for the ileus, but we will try to figure out a way to do that. Thank you.
Gail Besner from Columbus, Ohio. That was a really elegant and beautifully presented study, so congratulations on your work. A number of years ago, we also looked at the enteric nervous system in histologic specimens resected in the operating room for necrotizing enterocolitis. These babies typically get a stoma, and so we were able to look at specimens during the acute neck period and then several months later when the stomas were closed. And as you showed, there are abnormalities in the enteric nervous system during the acute event, which you can completely understand because the intestine is severely injured. But interestingly, even months later, when the child is perfectly healthy and you're closing the stoma, there are still absolute abnormalities in the enteric nervous system. So I think you're onto something really, really important here. And so the question, my question is, you want to be able to treat the patient perhaps with pro-motility agents at the right time, but when is the right timing for that? And how would you envision this being used clinically in terms of when you would administer these types of therapies? Congratulations.
Thank you, Dr. Besner, and thank you for your leadership on this work of the enteric nervous system and neck. That's a fascinating paper when you looked at the stoma closures as well as the initial resection. So to get to the question, I think that it's not necessarily going to be using these prokinetic agents because their safety profile is not without risk, but the thing that we're looking at now is to determine what components of breast milk lead to enteric nervous system maturation and improved motility, because we know that breast milk babies have better motility even when, not compared to neck babies, but compared to formula-fed babies who don't develop neck. And that might be a really safe and productive way to encourage enteric nervous system maturation, avoid neck, and then that could be included to improve our formulas. Thank you.
Augusto Zani, CKIDS Toronto. Again, very elegant. My question is regarding something that has come out very recently from your lab and that we've been interested in, which is in fact brain damage secondary to necrotizing enterocolitis. So we've shown, and other people have shown that there is a damage of the central nervous system. And now you're looking at the peripheral nervous system. And so I was wondering whether you can elaborate how you think, whether these two things can go together. We really don't know how. We talk about the gut-brain axis. Do you think that the injury to the ENS could be actually part of that gut-brain axis that can then activate the microglia and the astrocytes in the brain?
Thank you for that question. It's certainly an area that our lab is focused on. I think that the potential connection might be in a special population of cells that communicates directly between the enteric nervous system and the central nervous system. Yeah. And so we're looking at small populations of cells in the intestine that do that. And they have been shown to have a connection in like IBD, for example, between the central and the enteric nervous system. So I think there could be a connection there. Thank you.
Thank you.
Professor De Coppi.
Yeah, De Coppi, London. Thank you for a very elegant study. And just a quick question related to if you think that this could be an innate immune response actually related, and you've shown before TL4 and so on. But have you looked at the ENS distribution in other part of the gut, for example, in the esophagus or in the stomach, that are not involved in the NEC, so to see if that is more of a systemic response?
Thank you for that question. That's not something that we've done yet, but it's certainly a great idea and something that we should do. In our model of the disease, most of the inflammation is seen in the small intestine, so that's where we focused our attention, but we haven't looked at the ENS and the other parts, but we certainly will take that into consideration. Thank you.
Thank you.
Well, thank you very much, Mark, and congratulations for your work.
Great job, Dr. Kohler. I'd like to bring to the podium Dr. Sho from the Hospital for Sick Children, University of Toronto, for his paper entitled Tetrahydrobiopterin ameliorates experimental necrotizing enterocolitis by reducing the oxidative stress. Co-authors, as you see, include Drs. Mayaki, Alganabi, Jansen-Locke, O'Connell, Lee, and my co-moderator, Dr. Agostino Pierro. Please.
Good morning. Thank you for this opportunity to present our work. Today, I'd like to talk about the effect of tetrahydrobiopterin on experimental neck. We have nothing to disclose. As you heard before by previous speakers, neck is one of the most severe gastrointestinal disease affecting premature babies. The incidence and mortality remain high, indicating the need for innovative treatments. Pathophysiology of neck is still incompletely understood. Recently, oxidative stress has been reported to play an important role in neck pathogenesis. Our collaborator reported that tetrahydrobiopterin, or BH4, present on high concentration in human breast milk and no BH4 in formula. BH4 is a cofactor of nitric oxide synthase, regulating nitric oxide production. This may explain the protective role of breast milk in neck. As I presented now, BH4 is a cofactor of nitric oxide synthase. BH4 levels are influenced by oxidation, forming catalytically incompetent BH2. Both BH4 and BH2 bind off nitric oxide synthase with equal affinity. But BH2-bound nitric oxide synthase generates superoxide rather than nitric oxide. In addition, BH4 is a scavenger of reactive oxygen species, reducing oxidative stress. This is our hypothesis of the role of BH4 in neck development. Oxidative stress during neck development changes BH4 to BH2 in the intestine of premature babies. The superoxide derived from BH2-bound nitric oxide synthase give rise to more oxidative stress in the intestine, forming more BH2. This feedback loop may be associated with neck pathogenesis. So we hypothesized that exogenous BH4 administration scavenges reactive oxygen species, reducing the oxidative stress. Less oxidative stress leads to less BH2 production, which results in less superoxide. This effect may block the negative chain of neck development. The aim of this study was to determine the effect of exogenous BH4 on experimental neck. We've been using the animal neck model described in 1975 by Dr. Barlow using hypoxia and garbage feeding of hyperosmolar formula. In addition, we added lipopolysaccharide administration. There were 3 groups in this study. Breastfed pups served as control. Neck was induced by the 3 factors. Mouse pups in the BH4 treatment group received intraperitoneal injection of BH4 once daily during neck induction. Terminal ileum was harvested on postnatal day 9. First, we looked at the intestinal oxidative stress using T-bars. The intestinal oxidative stress was increased in neck compared to control. On the other hand, it was decreased in the BH4 treatment group compared to neck alone. There was no difference between control and BH4 treatment group. Second, we looked at neck-like intestinal injury, including histological score, intestinal inflammation, and intestinal hypoxia. In control group, intestinal villi and epithelial were intact. In neck, you can see low villus height, epithelial sludging, and disarrangement of villi. When we gave BH4, the neck-induced alteration was reduced close to normal. Histological score of neck severity was decreased in the BH4 treatment group compared to neck alone. Similarly, intestinal inflammation was decreased in neck compared to control, as indicated by pro-inflammatory marker TNF-alpha and IL-6, whereas the markers were decreased in BH4 treatment group compared to neck alone. The 3 genes are hypoxia markers. These were increased in neck compared to control, whereas these were decreased in BH4 group compared to NEC alone. Again, a possibility to explain the mechanism of action of BH4 administration in our findings is shown here. Exogenous BH4 scavenges reactive oxygen species, reducing oxidative stress. Less oxidative stress leads to less BH2 and less superoxide. This effect prevents NEC development we talked about earlier. In conclusion, BH4 administration during experimental NEC reduces oxidative stress, severity of the disease, and intestinal inflammation. For potential translation, BH4 administration may prevent the disease progression by reducing the production of reactive oxygen species. Thank you for your attention.
Thank you for that really elegant presentation. Beautiful data, as always, from Agostino's lab. Please.
Simon Eaton, London. Very nice data. And I just want to make a comment here, which is actually just speculation that could tie together the two previous presentations, so the one from Dr. Hackam's lab and the one here from Dr. Pierro's lab. That is that tetrahydrobiopterin isn't just involved in nitric oxide synthase activity, it's also a cofactor in the production of serotonin in the gut. So actually your serotonin here could be actually also improving gut motility, and so there could be a link between this in the mechanism of the protective action. I think that's possibly an interesting area to explore.
Thank you for your comment. I haven't looked at the serotonin, so it is a great idea to reveal the mechanism of action. Thank you.
Sure. Thank you. Other questions? Very quickly, as our next presenter perhaps could come to could make his way forward. So oxygen species are important in the pathogenesis of the disease, as you clearly show, but they must have some protective benefit in terms of controlling inflammation or even controlling bacteria. Any thoughts on potential downsides of this approach?
So I think BH4 also affects nitric oxide production. So this may improve nitric oxide production from enos. This may prevent NEC development. On the other hand, BH4 induce excessive nitric oxide production from inos. So this leads to tissue damage. That's why I think we need to give BH4 in early stage of NEC before inos application.
Perfect. Thank you. Beautiful work.
It is a great pleasure for me to introduce the next speaker, Dr. Simon Eaton, because I've been collaborating with him for many years in London. And Simon is going to present today a paper on the mitochondrial DNA is elevated in plasma of infants with necrotizing enterocolitis and acute abdomen. And this is a paper which is talking not about experimental NEC, but talking about what's happening in humans. Thanks, Simon.
Thanks, Agostino. So this work was performed by a master student, Georgina Navoli, in this laboratory, and she's the first author of this paper. I've set the following disclosures and conflicts of interest. So there's a recent terminology that's become quite frequently used, which is that of DAMPs. DAMPs are damage-associated molecular patterns. And mitochondrial DNA is an example of one of these damage-associated molecular patterns. So the mitochondrial DNA actually looks more like bacterial DNA than mammalian DNA. And so actually it can get recognized by systems that look for bacterial DNA. And so if mitochondrial DNA is released from necrotic cells, it can be recognized as bacterial by TLR9 and therefore trigger neutrophil infiltration, extracellular trap formation, and might therefore be found in netosis. And these are all increasingly being recognized as potential mechanisms of tissue damage in NEC. So mitochondrial DNA outside of cells could be a marker of necrotic damage, but could also propagate the inflammatory response both locally and systemically. And recently, cell-free DNA, mitochondrial DNA in plasma has been shown to be elevated in chronic diseases. So on the left-hand side, you see elevation in rheumatoid arthritis, and on the right-hand side in coronary heart disease diabetics versus diabetics without coronary heart disease. Not only in chronic disease, but also acute diseases. So in trauma, an elevation of plasma mitochondrial DNA, and also there on the right in sepsis. So we hypothesized that cell-free mitochondrial DNA would be elevated in the plasma of infants with conditions in which there's intestinal necrosis, such as NEC, and acute abdomen, such as volvulus, etc. And so we undertook a proof of concept pilot study with 4 groups of samples. These are control infants less than 32 weeks gestational age with no suspicion of NEC or sepsis, infants with Bell stage 2 to 3 NEC, closer to term infants with acute abdomen, volvulus, etc., with suspected ischemia, and also infants who are undergoing an operation for things like atresia, where we wouldn't expect ischemia. And so from plasma, because it's usually thought of containing no DNA because you've got no cells, then actually isolation of cell-free DNA is quite technically challenging. And so because it's in very low amounts, so we use a specific kit that's needed to isolate. It requires 200 microliters or more of plasma. And actually it's very difficult to actually quantify the amount of cell-free DNA that you get from a plasma sample. What we then did was some colleagues of mine who are better at molecular biology than me isolated the mitochondrial DNA for mitochondrial CO3, which is one of the genes encoded for in the mitochondrion, put it in a plasmid and then expanded it. This enabled us to make a standard curve of mitochondrial DNA by copy number. So here you see the relation between cycle threshold number on a quantitative PCR assay with the number of copies of mitochondrial CO3 gene. So then when we look at our actual data, what you see here is the premature controls compared with all 3 surgical patient groups. So that's the NEC, the acute abdomen, and the laparotomy with no suspicion of ischemia. You can see 2 things there. Firstly, that the results are expressed on a logarithmic scale, and that there's still significant outliers. So most of the other studies have also shown a very wide variation, so quite often express data on a logarithmic scale. However, we are slightly concerned that the significant outliers might be due to things like not having a good plasma sample due to hemolysis, etc. So we're slightly cautious about those large outliers. So a highly significant difference between the controls and the patient groups. When we compared the premature control infants, non-surgical, with the term infants undergoing laparotomy or laparoscopy, there was a significant elevation. We can't tell because we don't have the appropriate samples to tell whether this is due to age or due to the surgical condition that these patients are necessitating a laparotomy or laparoscopy. When we compared plasma mitochondrial DNA in NEC infants compared to their premature age-matched controls, there we saw a significant elevation. What we don't have the data from, because this was a multi-center study, is the data on the extent of the necrotic bowel. So in the future, we'd like to take this forward by looking at to see whether the amount of mitochondrial DNA in plasma relates to the degree of necrosis, and also does it relate to parameters of systemic inflammation. And then finally, in the patients that appear to have volvulus or other conditions of acute abdomen, there was, again, a significant elevation. We don't know whether this relates to the amount of necrotic bowel. So in conclusion, plasma mitochondrial DNA is significantly elevated in NEC and acute abdomen. Further work is necessary to refine the method to make sure that we're getting a good prep here of mitochondrial DNA that's free from artifacts. And we need to know that whether plasma mitochondrial DNA is a biomarker for the extent of necrotic gut and does it drive further inflammation. Thank you very much, and be pleased to take any questions.
Okay. Simon, thank you very much. This was fantastic. We can allow one quick question because we have the following session started right now. Is anybody... Okay, maybe what we could do is that you could meet Dr. Eaton outside and ask for more questions. I'm sorry, Simon.
That's okay.
And I would like to thank everybody that contributed to this fantastic mini symposium on NEC, going from experimental design to human results. And thank you to the presenters and to the people in the audience. Thanks. Bye-bye.